Okay, cool. Well, thank you guys for all coming to DeSciNYC here, and it's a pleasure to have you here. And I'll just tell you a little bit about what is DeSciNYC. It's a monthly meetup that we talk about all different things about the intersection of blockchain and science. And we get a lot of people. We have a website. It's about speakers, it's about community, it's about pizza. We record all our talks and make them accessible online. We have a group chat application here where you can have a little, after the party is over, you can still continue to chat with people. So you can fill that form out. Cool. Here's some of our previous speakers. We started off with a distributed mushroom collecting, and then this guy who was doing self-administered psychedelic experiments, and then someone who talked about bees, and then we had Martin Shkreli with AI in medicine. We had the guy that invented the NFTs. We had the person from Research Hub who started that. We had this Psi Combinator demo day. We had people on longevity from CryoDAO, from Age One. We had a professor from Albert Einstein Medicine on longevity and tissue replacement. And if you guys took pictures, make sure to tag us on Twitter or Instagram. It's really, we love it when people retweet. It sort of sucks to have to beg for it, but whatever. It was very helpful if you follow us, and if you took pictures, I'll retweet them and would greatly appreciate it.
Can you send this point out afterwards so we have?
Yeah.
Are the talks recorded?
Yes, should be here. Is it recorded?
Yes, it is.
Okay.
Where do you put them?
On DeSci, DESCI .NYC. Okay. So take pictures, tag us. What is decentralized science? It's like the emergence. So decentralized science came from sort of DeFi. And DeFi was everyone was trading tokens, everyone's creating tokens and exchanging value. But what's really cool about DeSci is that people are trying to use the blockchain instead of just sort of swapping tokens. People are adding data on-chain. People are doing analysis collaboratively. People are splitting up the IP between people that started projects. And so there's exogenic information coming into the blockchain. And then we're using this distributed ledger technology to try and make it more of a meritocracy around who's credited for what, and incentivizing scientific research in new ways. And there's all sorts of reasons why you want to do this. Traditional science has a lot of problems in funding. There's only so much funding to go around, and it's typically granted by a government agency. And decentralized science offers a way to crowdsource the funding. Peer review can sometimes be slow, and distributed ledger technologies can offer a way for people to crowdsource peer publications. And I think one of the more interesting things is allowing people to incentivize science in new ways. So currently science is incentivized using sort of academia, and you have to get a postdoc, or you have to get a professorship, or you're trying to get into a good graduate school. And then you didn't start a company, but all these sort of incentives can sometimes be a little bit misaligned and create sort of amalgamations that we weren't expecting to begin with. What's cool around decentralized science is when you start a project, you're creating sort of a company, and that company holds IP, and then everyone that sort of participates in the company, it gets tokens in reward for the data that they contribute, in rewards for the research, in the rewards for the numerical analysis that they do work writing it up, and those tokens can then be used to buy whatever, or as a status, or for lots of different things. So it makes the beginning to the end process of doing science a little bit more open and a little bit more clear from the get-go. So I'll just talk about, and then for our next talk, so we're going to do another one, and this one came out relatively quickly. So this one's on February 22nd, and we have the founder or the president of the Enhanced Games. So the Enhanced Games are a really interesting idea of using like the Olympic type sports in competition. And it's like the Olympics, but instead of the Olympics being where they check people for doping very carefully, it's like there's no checking. You're supposed to dope. And what I think is interesting about it from a scientific perspective is it's like a way into looking into how performance-enhancing drugs can help with medicine or help with sports. And right now, medicine is a little bit retrospective looking, where it's like someone gets sick, and then they come into a doctor, and they're like, okay, we have all these different things. But what's interesting about this is they're taking a healthy person and either making them even more sick and get cancer at 25, or trying to enhance their life and maybe prevent death, give them more muscle mass, and sort of seeing what happens. So I think it'll be interesting to see what happens, where they go with this, and to look at it from the point of view of like a science perspective. So the link to do that is here. Does anyone have any questions around decentralized science? Yes.
I guess, would you mind comparing and contrasting it to archive?
Yeah, so archive is around the final publishing of it, and there you're still not, you're not getting any, like you've done all the research, it's been funded by the government, it's been funded by, the research happens within a university through a PI, through graduate students, through research assistants. They then send out surveys to people that don't necessarily care about taking the survey and are just trying to get through it as quickly as possible to get their $20 Amazon gift card. And then they go take all the research up and write it and publish it in an archive where there's either no peer review or open access quickly, but you can't see like necessarily who contributed what to the science project. And if you contribute, you don't get any sort of credit beyond like your name at the top of the paper. But it is a good step in the right direction. Whereas maybe a decentralized science approach would be someone comes up with a hypothesis that people can participate in, and then multiple people add their data to the blockchain, so there's provenance for every part of the data. Everyone that contributes is part owner of the IP and equity of the project, and then they can sell their IP or equity for money, use it as a way to get a job, or any other number of things. Anyone else? One more. Yes?
I feel like my question with decentralized science that I always wonder is like, when citations are the primary driver of funding currently, what do you think would incentivize like a promising young scientist to say, I'd want to contribute to a decentralized project if it potentially like takes away from the method of growth of their career currently?
Yeah, I mean, I think you can make more money in decentralized science because you might be limited to a certain salary. And so there might be a possibility of making more money in a unbounded way, where you're sort of working more for yourself as opposed to working within a traditional academic setting. And of course, it comes with you can make no money as well. And it's for people, I think it's not trying to compete for those types of things, honestly. I think it's for people who are looking to go a separate path in trying to do research as opposed to trying to compete with things. So like decentralized science projects aren't the type of thing that people aren't trying to do like basic research right now. Whereas in like traditional universities, there's a lot of basic research being done, but there's also a lot of non-basic research being done in universities, which I think can be better done in a decentralized science approach than through the traditional academia. Cool. Okay, so we have two amazing speakers tonight. The first is Vincent, who is a professor at Columbia University in the Neuroscience department. And he's been doing this very interesting thing, which we're going to talk about, on Focused Ultrasound. And Focused Ultrasound has been, as of late, become very, very popular because it is completely non-invasive, and it's pretty generalizable around the body, and very cheap to administer. So he has been doing this for 12 years. So we're excited to hear what he has to say. Thank you, and we thank you for coming out tonight as well.
I think it might be better place now.
Yeah. Let me just make sure it works if I have to get a stage. Okay, perfect. All right.
Okay, great. Well, thank you very much for inviting me, and thank you all for coming out. This is really exciting. I fully support decentralized science. We, we're putting a lot of data sets on the internet and, publishing on bioRxiv and stuff like that. It's a really great concept. And, freeing ourselves from the shackles of traditional science, can only benefit everybody, I think. Also, I have to say, this is the first time I've ever given a talk in socks. And that's perfectly fitting because this is going to be a little bit casual. So, bear with me a little bit. So, yeah, that's me. I'm at Columbia. My disclosures are, I'm just a poor scientist. I have no financial relationships. Who is this guy? So I'm a cognitive neuroscientist. I'm interested in learning, memory, attention, decision-making in prefrontal cortex and in the basal ganglia of primates, which includes monkeys and humans. The tools that I use heavily rely on behavioral methods, electrophysiological methods, these neat new neurotransmitter reporters, which is a whole other story that I'd love to tell you about, but no time. Chemogenetics, which you might know something about. And what I'm going to focus on, no pun intended, is focused ultrasound. And so, at this stage in my life, I'm a full professor, my kids are grown, I'm an empty nester. I have a real personal interest in this topic of healthy cognitive aging throughout the lifespan, so from childhood to, cradle to the grave, as they say. And the field as a whole, I think, we love our tools, we love our optogenetics and chemogenetics and all this invasive kind of stuff and deep brain stimulation. But, we really, as a colleague of mine said, nobody wants a hole drilled in their head, right? And we want non-invasive technologies. And ultimately, 100 years from now, or, star date 2347, whenever it happens, we do want to get to a fully non-invasive kind of, Star Trek sickbay scenario. So that's, I hope, we're taking some small steps in that direction now. So that's a little bit what I'm going to talk about. So we're talking about ultrasound. What is ultrasound? Well, it's really any sound frequency above 20 kilohertz. 20 kilohertz is the limit of human auditory perception, and so we just sort of arbitrarily say that anything above that. But the kind of ultrasound we're doing is actually 10 times higher. It's in the 200 kilohertz to 2 megahertz range generally. And the way we produce that is we have a piezoelectric device that's embedded in this ceramic, it's basically just a shallow ceramic bowl. And this can do two things. One is it can produce very high frequencies, like I talked about, but also it can focus the ultrasound down to, we like to talk about the grain of sand, the grain of rice, grain of sand, whatever. So, we talk about the focus being about the size of a grain of rice. Maybe it's a little bit bigger, that's something we can talk about. So this is, different but complementary to the traditional imaging B-mode ultrasound that you're all familiar with. And so what does ultrasound do? Well, lower frequencies, so ultrasound can penetrate into the brain, and the reason it can do that is because the pressures at the face of what we call the transducer, so that little bowl-shaped thing I showed you, we call it a transducer. The pressures at the face are very low, and then the pressure increases as you get towards the focus. We can vary the frequency. Low frequencies have deeper penetration, but a bit, the focus is more spread out. Higher frequencies have less penetration, but a smaller, tighter focus. So we tend to use higher frequencies in smaller animals like mice, and lower frequencies in primates and humans. What are the effects on biological tissue? Well, basically, there's a mechanical effect, of course. Its sound is a pressure wave, and it's causes vibration, as we all know. there's also a thermal effect. So there's heating at the focus because of the energy that's being deposited. And something that you probably might never have heard of is something called the radiation force. So the sound wave actually pushes and causes a displacement of tissue, and that's something that you can actually image. You can image it with MRI. You can also image it with ultrasound itself, like I said, on the B mode imaging. And so the displacement can give you sort of a readout of the pressure wave inside the vessel, in this case the skull, that we're introducing it. And so that's really exciting because, I mean, we do have this thick skull to deal with. It's a bit of a problem, but we do have this sort of crazy idea that we can have a completely self-contained system where we have focused ultrasound going in and stimulating, and we can read out with imaging ultrasound. This is sort of cutting edge stuff. But this field goes way back. So the earliest study that people generally refer to was by Harvey in 1929, almost 100 years ago. And he just did the Luigi Galvani experiment, basically, of causing a frog leg to twitch with ultrasound. And then he showed that you could block that with curare, so it was mediated by the nerves. Then about 30 years later, Fry et al. Did an experiment in a living animal where they focused ultrasound into the brainstem, in particular the visual thalamus, and they were able to measure electrical potentials on the surface of the brain, the visual cortex. Okay, so that's some evidence that the ultrasound was actually stimulating the thalamus and stimulating the fibers that go from the thalamus to the cortex and causing a response. And now we can actually induce what are called phosphines. A phosphine is just a flash of light that you see, sometimes when you're lightheaded or when you get hit on the head. That can be induced with ultrasound. So I'm going to talk about basically, if you look at the field, there are basically 4 ways that ultrasound is being used. Neuromodulation, which is a direct mechanical or thermal effect that directly stimulates the brain. Blood-brain barrier opening, which I'm going to talk about, where we combine ultrasound with intravenous microbubbles. Then there's thermal ablation, which is a purely thermal effect and is actually FDA approved as a treatment for essential tremor, but I'm not really going to talk about that. A lot of medical centers now have these devices and are doing ultrasound ablations as a service. People come in with essential tremor, they have the procedure, they walk out without essential tremor. It's really amazing, but I'm not really involved in that research. And finally, functional ultrasound is a way, it's a novel imaging modality where you can actually image blood flow. So you can actually image, like an MRI, you can image the function of the brain in real time. And people are using this in really cool ways, like to read out, movement and tension and things like that in the parietal cortex. But I'm going to talk about mainly these first 2. In terms of the biophysical mechanism, people are interested in these TRP channels, which are present in the brain and are temperature sensitive and probably have something to do with the response. We don't really know a lot about this, or I should say I don't know a lot about this. It's not my area of research. But this is something to keep an eye on in the future. This is what got Artem Patapoutian the Nobel Prize a couple of years ago. Okay, so I'm going to talk mainly about, not biophysical, but the whole other end of the spectrum. I'm going to talk about behavior in primates and how that can be modulated with ultrasound, and specifically, how we can, change their cognitive performance on certain tasks. So I'm going to show you. So this is the setup. We have a monkey, and he's interacting with a touch panel, and he's doing a little task. He touches the panel. And he gets apple juice when he does it right. He doesn't get apple juice when he does it wrong. That's very motivating, and he loves to do that. And he'll sit there for a couple hours every day. And so our first question was, okay, if you have an animal, an awake animal, what is their reaction to ultrasound when you apply it to the brain? We didn't know. Anything could happen, right? I can't even go through all the possibilities. So the first thing we wanted to see is the first question is what happens? Really just pretty basic. So we've got him doing this task, and I'll show you exactly what he's doing in a second here. Where's my mouse? Oh, there it is. Okay, so here's a little movie. And so there's these two motion patches, and one of the patches has motion that's a little more coherent than the other, and you can sort of sense what the direction is. So this one is moving coherently to the right, and the other one's just kind of random. So he has to pick the patch that looks like it has more coherent motion. And they get really good at this, and you can make the task easier and harder just by varying this parameter. Okay. Next slide. We're targeting the basal ganglia specifically. So this is a slice like this. It's an MRI slice, what we call a coronal slice. This is all cortex. It's not a great MRI, so I apologize for the low contrast. This is what we call the striatum. It's a part of the basal ganglia that we think is involved in learning, attention, memory, and decision making. And it's divided into two structures, the caudate nucleus and the putamen. And that's as much neuroanatomy as you're going to get. So, and then this is a simulation of the pressure wave inside the brain that we're hoping to produce. So the transducer is sitting here, and this is the pressure. And so here we're targeting the structure called the caudate nucleus, and here we're targeting the putamen. And so, so the subject is sitting and he's doing this task, and we give him a few minutes to warm up. So this is kind of like the warm-up period, and he just gets into a rhythm of hitting the touch screen and getting his juice. And then this is where we turn on the ultrasound, and we turn it on just for 2 minutes. And this is at 500 kilohertz, and it's pulsed. So we don't want to, we don't want to cook his brain, and we're using a low pressure. So the pressures are on the order of 200 kilopascals to 1.2. So what's a Pascal? 1 Pascal is 1 Newton per meter square. 100,000 Pascals is roughly 1 atmosphere for people who are into physics. Okay, so that's as much. It would translate into 194 decibels if you could hear it, but you can't because it's way too high. But that's like a rocket launch, basically. So it's a pretty high pressure. And so we're worried about what it's going to do to their brain, obviously. But the animal started working. We turned on the ultrasound for 2 minutes. It didn't bother him. He just sat there, punching the screen and doing his thing, as if nothing was happening. And we checked, to make sure that we actually turned the thing on, and did all the kind of sanity checks you would do, and we measured the output and this and that. And indeed, the device was on, but the animal kept working. And not only did he keep working, but he actually got better and better. So this is just showing his response time, and his response times are actually getting faster after we turned on the ultrasound. So that was actually a good sign. It was like the first thing we ever did with this. And we were, like I said, we were really worried. We didn't know what was going to happen, if there would be some weird reaction or whatever. So this is a good sign. And, we felt we were kind of on the right track. This task gives us a lot of things we can measure. So we can look at motivation in terms of response times and things like that. And we found that the ultrasound actually improved his motivation when we turned it on. We found that his performance was better in terms of he had more correct responses and he was faster at producing those responses. So we were enhancing his processing speed a little bit by turning on the ultrasound. So that was interesting. That's promising. We did this experiment. So clinicians always, get on my case because basic scientists like to do, they like to do proof of principle and just get stuff, published as quick as possible. And long-term studies just aren't done, and clinicians really hate that because, it turns out, stuff will get approved for human use, and it turns out the long-term studies were never done, and it's a problem. So we did a long-term, over a period of 2 years, we did 140 sonications in 2 animals, and then we looked at their MRIs, and their MRIs were as if they had never been treated. Basically, there's no difference in the pre and the post MRI after 2 years and 140 of these treatments. So again, that's a good sign. It means we're having an effect on behavior. We're not destroying the brain, and that's kind of important if you're going to do this stuff in humans. We're also interested in memory. So here's a little memory experiment, and see if you can figure out what's going on here. I'll give you a minute. Okay, so did anybody get it?
Yeah, it's what shapes stay throughout each trial.
Right. So there's an array of shapes, and then there's a little delay, and then another array, and one of them has changed, and you just have to figure out which one changed. And it turns out that's a fairly effective way of measuring what's called working memory or short-term memory. So we did that, and we had a control condition where there was no memory requirement. And these are the controls, the shams without ultrasound, and this is the error rate as a function of how long of a delay there was between the two things. And we measured the baseline error rate, and then with ultrasound, and we found that the error rates went down, actually in both conditions. So we think that possibly there could be some benefit to short-term memory of doing this.
Do you know the underlying cause? That's is it increasing the blood flow in the area, or
So we think it's basically just stimulation. So we're actually doing experiments now to compare ultrasound with basic deep brain stimulation.
Got it.
And so we think, but we don't know. We haven't really measured the neural activity, which is something we have to do. But when we get to the end, I'll show you another possibility, which is that we're actually altering functional connectivity in the brain.
Okay.
Which we've shown with MRI. So the next thing I'm going to talk about, so that's just ultrasound alone. Next is we're going to combine ultrasound with microbubbles and use that to open the blood-brain barrier. And the way it works is that we have these microbubbles. They're little lipid spheres. They're filled with a gas, and they're about 5 microns in diameter. 5 microns turns out to be about the diameter of a capillary. And so when they go through the capillary and they interact with the ultrasound, they just oscillate a little bit. And that oscillation puts a little bit of pressure on the capillary walls and makes the capillaries a little bit leaky. Because the capillaries in the brain have very tight junctions in their endothelial cells that prevent things things that are dangerous, like poisons and toxins and viruses and so forth, from getting into your brain. But we can use this method to make the capillaries a little bit leakier. This was first done in mice, and then my lab, I think, was the first to do this in primate model. But now it's done in humans and so forth in their clinical trials and so forth. This is actually showing, the resolution isn't great here, but this is a little tube filled with microbubbles, and this is showing the ultrasound pressure. And you can see when the pressure comes on, right there, you can see that change. Did you see that? Okay. So we'll go one more cycle. Okay. Ultrasound comes on there, and you can see the little bit of expansion of, this is just a plastic tube. It's not a real capillary, but that's the basic principle. Yeah? So again, the question is it actually safe to do this, or does it even work at all in a large animal? I mean, it works in mice, but mice have a very thin skull. Humans have a fairly thick skull, and not only do we have thick skulls, but every human has a different shape and thickness to their skull, and so there are a lot of challenges there. So again, this is the pressure wave simulated, and then we combine this with the microbubbles, and what we found was that we did a contrast enhanced MRI, and we found that the blood-brain barrier was permeabilized a little bit in the region of the highest ultrasound intensity. And then that's here. We've done this now, I want to say hundreds of times. We've done it a lot. And it's extremely reliable, and one of the main things that we're working on is just making it more precise and controllable. Anyway, this is just showing, this is a penny. These are some openings, and you can see the size. So we can control, we have pretty good control over the size, by varying the pressure. We can, choose different targets and so forth, and we can construct, actual physical models of the pressure wave inside the head and predict what's going to happen.
Do you ever try and target like density as opposed to volumes? So if you have like a resonant frequency, you could possibly oscillate some density.
Yeah, it's an interesting idea. So it tends to work better in gray matter than white matter. We don't know exactly why. It could just have to do with the amount of capillaries, or it could just have to do with the structure of the tissue. But I mean, it's a good point. We need to have simulations that take into account the variations in the structure, in the actual tissue structure.
Got it.
And whether it's fat or neurons or whatever.
And you could like target a certain like type of thing, right? Like if you hit the resonant frequency, if there was like a piece of paper is going to have the same resonant frequency as like as a spring or something.
Yeah, it's an interesting idea. I have to think about that more. How we can do that. It's I don't know if the tool is really that precise.
Okay.
Anyway, people ask how long does it stay open, so we did an experiment, and we measured, we opened on day 0, and then we measured days 1, 2, and 4 after that, and it took about 2 days for, so when you open the blood-brain barrier, actually you're causing the vessels to become leaky. You're also causing a little bit of inflammation, but that seems to close up within about 48 hours. Which is good and bad. I mean, it gives you a window, sort of a therapeutic window to work with, because you're going to have the barrier that's, it's open for a period of time where you could, do the procedure, take the patient back, to their hospital room, and then give them drugs and really monitor closely what's happening. But it doesn't stay open indefinitely. So that means that, if you were going to do this therapeutically, you'd probably have to do it repeatedly, and then that raises some real safety issues that have to be dealt with. Again, we did this, and we didn't know what was going to happen, in an awake subject, but we did it, and we got the same result, that in this case, we could apply the ultrasound, and we could give the microbubbles intravenously, open the blood-brain barrier, and it didn't disrupt the animal's behavior at all. In fact, the animal's performance was a little bit better on days when we disrupted the blood-brain barrier, and we don't know why. But they don't react to it. It's not painful. It's not it doesn't cause disorientation or anything like that, which is all good things and stuff that we really needed to know before we started doing this in humans. And then, so in terms of mechanism, I mean, we're really just scratching the surface right now. We don't know what's going on. I mean, when you open the blood-brain barrier, you could be changing oxygenation of the tissue, access to glucose, access to electrolytes. We're looking at these things using some MRI methods. So we're looking at oxygenation using something called QBOLD, which is a quantitative form of MRI to look at oxygen levels in the brain. But this is another finding that came up, which is that when you target one region, you can actually change the functional connectivity of the targeted region with other regions. So the ultrasound can actually, so functional connectivity it's a method to look at correlated activity using MRI. and you can sort of tease out networks and so forth. And so the ultrasound can actually change the functional connectivity. And I want to be clear, we're not changing the actual structural connectivity of the brain. We're not growing axons and things like that, but we are changing how remote brain regions interact with the region where the focused ultrasound was targeted.
How long does it take for the change to
we haven't actually measured the time course, but typically we'll do the ultrasound procedure, wait about an hour, and then do the MRI, and we can see the changes.
And if you don't stimulate again, do the changes stay?
Good question. We haven't done that experiment exactly.
Why wouldn't you expect some Hebbian idea of like the axons and dendrites firing together more intensive than they did before?
That's certainly a potential mechanism for this. That's a good idea. We would then want to go and actually measure that physiologically and see if that's the case.
Are the results path dependent? Like presumably there's some gradient of axon potential that's happening as the wave coalesces and becomes stronger. So if you target the same area from like different angles, like do you still see the same results?
Oh, that's a good question. We haven't actually tried that, but it affects different networks differently. We know that. We've published papers looking at the default mode network, the attention network, and so forth. So they're affected differently. This is early days, and actually figuring out what networks are affected and how they're affected and all that kind of stuff. There's a lot of work to be done. A long road ahead of us, but it's exciting. This is actually a connectivity map. I forgot what they call it, but and we've done this. We've shown changes in functional connectivity both with ultrasound alone and ultrasound combined with microbubbles. Another thing that we found was this is all done without drugs, and one thing that we're interested in is using this technique for targeted drug delivery. So we did some experiments. I'm not going to show the videos, but we did. We targeted the caudate nucleus. We opened the blood-brain barrier. We administered a drug called domperidone, which is a dopamine antagonist that's used for nausea. It doesn't cross the blood-brain barrier naturally. And so when we opened the blood-brain barrier, we saw an attentional deficit, which, we weren't that happy about getting a deficit, but it showed that the drug was working. So this antagonist could actually cause an attentional, it's called hemilateral neglect. And it's a very commonly seen after right parietal strokes. You'll have patients that sort of just ignore the left side of space. They don't orient to it visually. Their body representation has changed and so forth. And we saw a very clear transient example of that when we used domperidone. We also used a serotonin antagonist called ondansetron. Again, this is a nausea medication typically taken by cancer patients. When we gave it to the monkeys after the blood-brain barrier opening, they had a little bit of a neglect syndrome. And it went away after like 12 hours or something.
What is neglect?
So it's an attention syndrome where the subject basically doesn't orient to one half of space. So they will, if you show them a piece of food in the intact hemifield, they'll reach out and grab it. If you show it in the impaired hemifield, they have no reaction.
And this happened during and after the BBB opening?
Yeah.
Okay. So there was improved performance in some ways and then dramatically worse in others.
Right. But using these are antagonists, so presumably if we used agonists, we might get the opposite.
Yeah. Besides enhancing delivery of the drugs, in Alzheimer's disease, did you do any testing on delivery on drugs and also the antibodies?
Mm-hmm.
Did you do any tests on the antibodies as well?
So my colleague has a clinical trial on Alzheimer's happening now, and I think they're looking at some of those things. we don't really have a good primate model of Alzheimer's right now, so that work is mostly being done in humans.
Okay.
And I think, so I'm happy to talk about future directions. I don't know if I did.
Go for it.
Okay. So, the future of ultrasound in terms of research, I mean, there's just a huge number of avenues that are being pursued. My lab is interested in optimizing the efficiency and the safety of the method. We're doing, for example, targeting based on individual anatomy because, human heads come in all shapes and sizes and skull thicknesses. And we're actually using AI methods to try to predict how that, the skull is actually going to affect the pressure wave inside the skull. Something we're sort of thinking about, not really doing, but this is really cool. It's called acoustic lensing. And you basically 3D print something that goes outside the head, and then this can compensate for the shape and thickness of the skull. And there's this kind of idea that you could actually create like a 3D pressure hologram inside the skull. And so you wouldn't have a single focus, you would have many foci, and you could like really, get fancy with that. And that would be really fun to do. Emergent field of sonogenetics, which is where we combine, virally introduced mechanoreceptors, like the TRP channels, or there's a bacterial mechanoreceptor called MSCL. There's MEC4, which is found in C. elegans. And so you could introduce, this is not fully non-invasive, so there's a little bit of a downside, but you could introduce a virus into a particular brain target, have it express the mechanoreceptor, and then activate it with ultrasound. So that's something we're looking into. People are trying to do like visual prostheses with this, and creating patterns of activity in the visual cortex. It's pretty science fiction-y kind of stuff. In terms of commercialization, I think we're seeing more and more every year, more companies coming out in all the different, in the clinical arena. There's already the InsightTech device, which many medical centers are using and running a service to treat essential tremor. There are research systems that are pretty much turnkey. So if you want to do mouse research with ultrasound, Fus Instruments will build one of these for $50,000. And they also have an MR compatible system for $100,000 that will work with a, I sound like I'm selling this. I'm not selling. I have no relationship with this company. But they have an MR compatible system that'll work with a 9.4T system. And then some people are working on wearable ultrasound transducers. And, it's all very interesting. I think that we really need to get to something that's probably wearable, but also with multiple transducers so that we have more control over what's actually happening. So that's it. And I'm happy to take questions. More questions.
I have a question.
Oh, yeah.
Yeah, so with the slide, with the hemi-neglect, I'm just kind of confused because if you're targeting the basal ganglia, and if there's a parietal dysfunction where hemi-neglect would happen, how does that correlate?
Right. So attention, spatial attention, is controlled by many structures in the brain. The prefrontal cortex, the parietal cortex, the basal ganglia, and parts of the brainstem all control. So there's a nucleus in the brainstem called the superior colliculus, which controls eye movements, but it also controls attention. So spatial attention is one of these functions that's so important that a lot of your brain is doing it, including the caudate nucleus. And the caudate is also involved in controlling eye movements, so both overt and covert attention. Yep.
How did you decide on the frequency, duty cycle parameters for your ultrasound in the private experiments?
Really good question. I mean, we haven't really explored that space, and we need to.
Okay.
It's just what sort of happened to work, and it worked, and it's like.
Did you try a lot of things that didn't first, or?
We actually know, but there's no. I'm not saying that any of this has been optimized.
Okay.
So there's still, like I said, there's a long road ahead of us to actually optimize all of those parameters. There is this idea that certain frequencies cause excitation, and other frequencies and pressure regimes cause inhibition, and so we can sort of change the excitatory inhibitory balance by manipulating pressure and frequency. But again, early days, and all that really needs to be
I
explored.
saw some of the stuff from the Korean lab that said that from Korean studies that said that they knew which were excitatory versus inhibitory. Do you play stock in that?
Well, it's an interesting hypothesis. It needs to be tested more carefully.
Okay.
I saw a little bit ago that ultrasound was used to zap addictions away, which seems very promising use case. Would it be the same methodology, or was that just like pure science fiction?
I haven't seen that study. We tried to get a study funded where we were going to look at ultrasound targeted to the certain reward centers as a treatment for alcoholism.
Mm-hmm.
So chronic alcoholism is extremely refractory. As you know, if you have a relative or something, you probably know about this. So there was some interest, but we never really got that study funded. The idea was it's a structure called the ventral striatum, or the nucleus accumbens, and it gets a lot of dopamine.
Mm-hmm.
And people think it's involved in addiction and in alcoholism. So we're going to try that. Never got funded, so we never did the study. But I'd be interested in this other study.
So this is more about decentralized science and
Yeah.
So it seems like you have a massive search space that you could explore.
Exactly.
For this, and presumably limited resources. I don't know how many people you have in your lab, but like a few. Probably.
Yeah.
And then simultaneously, there are probably many other labs that have similar transducers and access to similar, animals that they can experiment with. What is the feasibility of parallelizing a lot of these experiments? Because even if you did have the resources, you would be limited by the sequential nature of like working in a single lab in a single place.
Exactly.
And human beings. And so, realistically, do you see any possibility of parallelization of these experiments and agglomeration of their results?
Yeah. So I'm going to say something politically incorrect. Not in this country, because that's just not the way the NIH funds science.
But if
it might happen in
not
China.
the NIH, what if for someone who wasn't the NIH?
Yeah, right. It might happen in China.
Oh, okay.
Because they do things big scale. It's no. I mean, what you want is something like the Allen Institute, but Allen Institute is focused entirely on rodents. But if we had an Allen Institute for human neuroscience, I mean, I think they do a little bit of human neuroscience there as well. But that's the scale that you kind of need, and that's you're talking about I mean, the entire NIH budget is about $50 billion, and to really do this at scale, you're talking about $1 billion or so.
So, and then what are the? I'll try to go quickly. What are commercially, how much money would there be to make, assuming these technologies were successful?
Yeah.
That's the question. How much? Would it be more than $1 billion? I guess. Would it be more than the cost?
Well, no. I mean, you can build these systems. You can build. A colleague of mine is building a phased array system. He's getting components from China, and it's about on the order of $50,000 per system. So if you wanted to have tens of these systems, say 50,000 times 10 or times 100, you know. So it's, you know, it's within the, you know, few million dollars, basically. You could have a pretty robust enterprise and start doing these studies.
So a lot of there's like like half of DeSci is like super bio-focused, and particularly around this, and there's a lot of focus around intellectual property, NFTs, and so I'm wondering if you had a bunch of scientists from all these different, because I know you're saying it can't be done by the NIH, and like I completely agree. That's one of the limitations that Michael was talking about earlier. But suppose, for example, that we focus a bit on the supply side, and we just have the scientists do these things anyways for free or whatever. But we kept a track their contributions to this project, and like basically allocated them a portion of the intellectual property that was created out of it. Do you think that it's realistic that because then again that intellectual property would have value, you know, probably very valuable maybe, and it could reward all the scientists who participated. Do you think that is possible in any way?
It's possible. You got some legal hurdles because the two institutions that are involved here are the universities and the NIH, and both of them have intellectual property interests that you would have to deal with. And so Columbia technically they own all of my intellectual property. And if I want to license something or patent something, I have to go through their lawyers. That's a real issue. I can't really do things on my own unless I sever that relationship. I don't know if people try to get around that. I haven't done that. I mean, I have in the past, you know, licensed some products through them, but it's, you know, it's an issue. As soon as you license something, NIH wants to know if that was supported by a grant, and then you have to go through their patent process and so forth. And you know, these are limitations, but you know, universities have a lot of power, and you know, and a lot of legal. I'm not going to say too much about it.
But I think that's why you need like DAOs to like fund initial research with like grants, so that people can have like the amount to live, but then also own the IP that would go into the grant, that would go into the DAO instead of having the IP go into like the IP licensing department of the university.
Right, right. But you know, changing that model is going to require, you know, a lot. I mean, I think institutes that are like fully self-contained, like the Allen Institute, you know, where you just have a, you know, a huge endowment, but they also get NIH money, and they also have university partnerships. So, you know, you have to deal with that at some level.
Yeah, you mentioned before that you are targeting viruses. So here's a practical experience. When people have a chronic BBPV, which is a recurring vertigo, they are trying to heal it with, you know, vestibular treatments. I had this chronic vertigo for 1 hour. Every doctor told me you're going to have it for the rest of your life. I did a lot of research studies on that. And I find out when you have the chronic vertigo, you have basically viruses that are bunkered in the vestibular nerve.
Yeah.
And you have to go after the viruses, not just mesmerize the vertigo with anti-vertigo drugs. So they give you, you know, a Valium, they give you clonazepam that is going to improve. Vestibular treatment with eplemenevir going to help that the crystals which get released there, they're going to vanish on. But the problem is that in the vestibular nerve, the viruses are recurring. So you actually target the viruses in order to get rid of the vertigo. So now your application is perfect for that. You can actually target the viruses in the vestibular nerve to see what is happening, and then you can use like acyclovir drug to go after them, you know?
That's an interesting idea. That's a very, anatomically, it's a very complicated structure because you've got the, you know, it's called the bony labyrinth, and it's embedded in the base of the skull, basically. So targeting ultrasound would be really pretty tricky there, but I see what you're saying. I mean, you can have vertigo from acoustic neuromas, from viruses, from the little ear stones, the otoconia get displaced sometimes, and there's sort of a maneuver you can do to getting back into place and things like that. I mean, ultrasound could potentially play a role there, but it's a very complicated anatomical structure, so it'd be kind of difficult. It's easier to go after things in the center of the brain, you know, because it's just a big melon. Question.
So for like the microbubbles, like why 5 microns?
5 microns is roughly the size of the capillaries. So you have a microbubble going through, and it's like just fitting through the capillary, and then you make it expand a little bit, and then you can, you know, get the capillary to you know, loosen up a little.
So could you potentially like make it smaller, or?
Oh yeah, yeah. I mean, they make these microbubbles in all sizes. They're originally developed, I think, as contrast agents for heart imaging. So when you get an echocardiogram and they want to do a flow study or something, they can use these microbubbles. So they come in all different sizes. We actually make them in different sizes, and then we use a filter. We call it a sizer, but it's a filter to select the 5 micron size.
Thinking about the like wearables and home use, how big is the, I guess, like margin of error for where you would put the device? Like if you have it off a little bit, will you still get the results that you want?
Well, that's why it would have to be packaged with a navigation system, you know, so you could sort of move it around and see in real time. You know, you'd have a computer that would show you in real time where the focus was, and then you could kind of like, you know, strap it down or whatever. I think in terms of the tolerance, it depends on, you know, what you're going after, what the structure is. If you're going after, say, like a target, like for, you know, the anterior cingulates, a particular part of the anterior cingulate is now a target for mood disorders and OCD. And that's a fairly large structure, so there's a good margin right there. But if you're trying to target the, you know, the acoustic, you know, the eighth nerve, that's you know, that's a different issue altogether. All right.
Why do the lower frequencies penetrate deeper than the higher frequencies?
It's just the physics. It's the same with light. Low frequency light goes deeper than high frequency when you're doing microscopy. It just has to do with the way it's absorbed and the, you know, the mechanical compliance of the tissue and things like that. It's a physics problem, and I'm not a good physicist.
Does age play a factor in this?
Yes, age is a very important factor and something that has to be looked at. And, you know, animal studies are almost always done on young, healthy animals, but if we're going to apply this, you know, to geriatric populations, we need to do, and that's a scarce and precious resource, you know, animals, because it costs money, right, to feed and house the animals. And so the older they are, the more expensive they are, or the more that they've cost you. So there aren't many places with geriatric populations of primates, for example. There are only a few places in the United States. But I think it's really important to do those studies before we, you know, develop this for you know, aging humans, of which I'm one. So we're all aging, just at different rates.
Okay. Excellent. All right. Is there any more questions? Well, thank you so much. Let's give another round of applause. Amazing. Okay. So next up, we have Siraj, and a little bio on Siraj is like a couple of years ago. I was doing the PhD in computer science, so I guess I was doing a lot of Googling on YouTube and just in general. Google knew that I was interested in AI. And Siraj would always come up on like the front page of my YouTube channel. And he has over 750,000 YouTube subscribers and literally a plethora of like any topic you could want, like whether it be Word2Vec, glove, Transformers, like any sort of topic that you could think of. He has like he's the modern day Donald Knuth with the art of computer science for YouTube. And now you're working on
sports betting AI.
Sports betting AI.
Yeah.
And so I mean, he's a great digital YouTube AI educator, and you guys should all definitely check him out on YouTube. He really has an amazing channel. So I'm very excited to hear your talk on AI and longevity and the sort of science experiments that he has been conducting. And I think you're also going to talk about AI intersection of science, which I think is probably one of the frontiers of science right now, because all the sort of research assistants, they could probably be replaced by AI in many ways. And we're just sort of scratching the surface on what does it mean for an AI to be a scientist. So without further ado, take it away.
Absolutely. Great. Thank you, Michael. Thank you, everybody, for being here. I know it's super late, but I'm going to try to keep everybody awake for the next 15 minutes, so stay with me. We have a lot of information to cover. In this 15-minute talk, I'm going to try to keep it at 15 minutes. We're going to first go over what is AI for longevity, very important topic, one of the hottest topics of the next decade. Then I'm going to go over my own journey in AI for longevity, and at the end, we, together, are going to build our own AI for longevity drug in 10 lines of Python code. All right, ready for this? All right, let's get started. So, what's the problem here? Everybody's dying, left and right, right? So the top 10 causes of death are all related to different systems in the body, whether it's the digestive system, whether it's the heart, whether it's the lungs, whether it's the brain. So you might be thinking that these are all disparate systems, that they're not interconnected, that they have nothing to do with each other. But the reality is that all of these diseases are interlinked in that they are all downstream of the root cause of all disease, which is aging. And that statement has gotten me laughed out of hospitals and clinics and different healthcare facilities when I say it, because this is a multi-billion-dollar industry, and I'm sure Vincent knows what I'm talking about, but this is something that people's livelihoods is against. They're paid to not say that aging is a disease, but in fact, Alzheimer's is a disease, and you should perpetually take this drug to cure yourself of Alzheimer's forever so that me and my shareholders make money like everything else in this country. But the reality is that aging is the root cause of all disease. And as radical a statement as that might seem, there are 3 different sources that I was inspired by to come to this conclusion. The first one is a book that I highly recommend you check out. It's called The Gene, and it's by Dr. Siddharth Mukherjee. And I got the privilege of working with Dr. Sid in LA at the LA Children's Hospital on a bunch of genomics data, and also for the U.S. Veterans Administration. And I got to see firsthand how incredible a time we live in it is right now, because we are seeing an explosion of omics data. Omics stands for molecular biology data. That includes gene data, so what encodes who you are, transcriptomics data, all these different omics. We'll get into that later on. But it's an incredible time to be alive, to see all this explosion in molecular biology data that did not exist more than a decade ago, now exists. And the second book is called Lifespan, Why We Age and Why We Don't Have to, by Dr. David Sinclair. Actually, people might know this guy. Raise your hand if you know Dr. David Sinclair. Yeah, he's gone on social media a lot, but this is a great book. He also has a podcast, highly recommended, the Sinclair Lab for Genetics. They've done decades of research. If you remember from the early 2000s, there was this craze around red wine and resveratrol. That came from their lab as well, but all that antioxidant stuff, it wasn't as impressive as what they later found, which is what I'm going to talk about. And the third, resource is my friend, Brian Johnson, who's been my friend for a few years now. We used to hang out in Santa Monica and talk about Kernel, which was his startup back in the day, which was a brain interface startup, and he was super into that. But nowadays, I think everybody here has heard of Brian now. He's working on longevity full time. And Brian and I have 2 different trains of thought here, which I'm also excited to go into, and what longevity means for you. So that's kind of my background. So before you and I get into building this in 10 lines of Python, we need to have some context here of how are we going to do this? How are we going to build an anti-aging drug in Python, in 10 lines of Python? It sounds ridiculous until you consider the context, in that we can consider everything information for a second. Just bear with me for a second. It's a little hypothetical experiment. Let's consider everything information for a second. Well, the most fundamental unit of information then is the atom. And what do we use with the atom? We build things. We build this desk. We build this computer. We build cars, trains, planes, all these things with the atom. And one of the things that we built with the atom was the computer. And the computer gave us a secondary unit of information, the byte. The byte is represented as a 1 or a 0, right? there actually happens to be a tertiary unit of information that people have been sleeping on, and that is the gene. It's not encoded as ones and zeros. It's encoded as A's, C's, T's, and G's. Who here has had your DNA sequenced before, by show of hands, just out of curiosity? It's actually a large number of people, given the privacy issues, which we're also going to go into. I've had my genome sequenced, and it's literally a .txt file on your computer. You can double-click on it, and it literally just looks like A, C, 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 T, 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 G. Literally everything about you is encoded in these numbers. And so that's just something to think about. Okay, so biology is information, physical matter is information, everything is information. Okay. So what evidence do I have here, other than these books that I've read and these people that I've talked to, that aging is a disease and that it's treatable? Well, 2 years ago out of Spain, there was this gene therapy that was applied to lung cancer patients. It was a gene therapy specifically for lung cancer, but as a side effect, some of the patients actually experienced age reversal. They experienced repigmentation of the hair, their wrinkles went away, so different body parts, X-differentiated is the actual term for this, right? Where X-differentiation is a term in the longevity community that means that different systems of the body lose their flavor or their distinction over time, which causes aging, whether it's in the epithelial cells or whether it's in blood cells, whatever it is. And these patients experience a reversal of it as a side effect. They did not expect that. But often in science, the most crucial discoveries are accidents, right? Watson and Crick, that's how they discovered DNA. And so my point here is that this is a wide open space. And if people are reversing aging by accident with gene therapies, then there's something about gene therapies that we should look at. Now, I am a computer scientist. Well, I actually dropped out of Columbia 3 years ago when I was studying computer science, as every Silicon Valley person should do, right? And what I came to terms with after dropping out of Columbia, I didn't really have an interest in biology when I was a student. What I came to terms with after is we should use all of this computer science knowledge to figure out ourselves and how we work. And that's what this explosion of biological data is going to allow us to do as people like myself who don't have any business being in biology. I don't have a degree or a PhD, but I will affect biology, and I think you can too. One of the ways that we can affect biology is by creating gene therapies. Gene therapies are essentially bits of information. Remember, all this is information designed on a computer. You and I can design this in Python or a different programming language. This information, once we design it, is then printed to a DNA sequence. Another way of thinking about this is that it is just like you would a 3D printer with PLA or some plastic. You can print out DNA material. And once this DNA is printed, it is put into a transportation vehicle like an AAV or some sort of viral vector or something, and then you could take it as a pill or as an injection or something like that. Now, if we just have a laptop and we want to hack on something, we don't have access to all this molecular material. We don't have access to a PCR machine or any of that. And that's where cloud bioinformatics comes into play. There are companies like Twist Biosciences that allow you as a developer to then push your code to the cloud and print out DNA that they will then send to you for you to test out on different organisms with, which we'll talk about as well. So where do we begin in biology? This space is super complicated, and as New Yorkers, one way that we can look at data and understand it is in terms of a metro map. What you're looking at here is a metabolic metro map. It is a map of all the different pathways in a system of the body known as the metabolome, and we'll talk about all the different ohms in a second. But as you can see, nobody should understand this from right off the bat. This is super complicated. And you can see all these different processes happening here, P-glycerates going up to the glycerol and the glycogenesis going downwards and all this stuff. This is super complicated. And entire researchers who I respect and admire dedicate their entire lives to one small part of this subway map. So it's super complicated, but what we have learned in the past decade is that there's 3 pathways in this metabolome that are super useful for longevity. Those pathways are, bear with me for a second, number 1, mTOR, which stands for mammalian target of rapamycin. We'll go into that. Number 2 is sirtuins, which stands for a subset of proteins dedicated to longevity. And number 3 is AMPK, which sounds like a punk band, but actually it stands for adenine monophosphate kinase, which is another longevity pathway in the metabolic pathway map. Now, why are these 3 important? Well, Dr. David Sinclair in his book, Why We Age and Why We Don't Have to, in his lab, he found that these 3 longevity pathways, when activated, actually reverse and stop aging in mice and in humans through these methods. Those methods include caloric restriction, exercise, resveratrol. Some of you might also know about cold water therapy, ice showers, all this stuff. What's the relationship between all of these different seemingly unrelated activities? They all cause stress in the body. It goes back to xenohormesis. That which doesn't kill you makes you stronger. And all of these things activate AMPK. So every time your body is under stress, it's like you're telling it you need to survive. You need to actually bunker down, take what little energy sources that you have, and have it last over time. And AMPK is also related to mTOR, which is also activated when you take too much protein. And protein actually increases your muscle mass. So when you're lifting, you actually feel great. When you have a lot of testosterone, you feel great. But actually, that's burning the candle faster. So you're actually going to die faster. I know that sounds kind of counterintuitive, but it's kind of like burning a candle too fast. And so these things are all interrelated. Now, these are just 3 pathways in that metabolic map. And they're just the first 3 that we've discovered. Another drug that is great for targeting AMPK is metformin. This is an FDA-approved drug. It was actually approved for diabetes, and as a side effect, helps people live longer. So there's something with insulin as well. So there's a lot of discoveries to be made in this field. Now, to me, let's get to me for a second. So what have I done here? I am just a mere mortal. Again, I don't have a PhD. I'm your college dropout. I'm your YouTuber. But I happened to reverse my age 8 years in 11 months, which actually beat my friend Brian Johnson in the Olympics, by the way. Don't tell him that. He's not going to like that. But the way that I did this is not by creating a list of supplements to sell on the internet. I'm selling a sports betting book. That's what I'm selling. The way I did that is just a simple tried and true method that has been working for decades. Diet, exercise, sleep, and healthy relationships / mental health. There's really nothing complicated about that, but what taking blood tests 3 times in 11 months did do for me that I measured on Inside Tracker, but you could do this on any website. I'm not saying Inside Tracker is the one you should use. What it did do for me is it showed me what I was doing and if it was good or bad. Like, for example, just, sleep more. Don't eat out so many times, like, a lot of sugar, or, don't eat burgers and fries every day, like simple stuff. I really don't think this is too complicated for people. Like, this has been going on for hundreds of years. Like, there's a study called the Blue Zones, right? And all of these places, Okinawa, Japan, Sardinia, Italy, Loma Linda, California, Nicoya, Costa Rica, they have nothing to do with each other other than the fact that they all live past 100 regularly. And their diets are totally different, right? You've got fruits and vegetables, you've got fish, you've got meat, you've got red meat, you've got dairy. Everything that people say that they are not into, these people have been eating forever, and they live longer, and they're happier than all of us. And so I think the reality of what we as mere mortals can do to live longer is to A, take blood tests regularly, and B, just try to eat whole foods, regular moderation diets. And this is just kind of a snapshot at my data at, some of the 42 different biomarkers that they measured: calcium, sex hormone binding, globulin, albumin levels. It's a liver protein. And what it does is it measures that level against a threshold that is, somebody of a given age, and it tells me the delta, like, oh, your levels are plus 1.2 years older than somebody of, say, 18, which is kind of the baseline. And what you can do is you can change your nutrients and mineral levels in your diet to try to make that number better. And look, we're all going to die. This is not going to fix anything: diet, exercise, sleep. That's just the reality. But it's a fun challenge, and it's a better way. It's a more holistic, preventative way of thinking about health and wellness. That being said, I do want to have a side note here, and that is to be wary, because the U.S. federal government is going to spend more on healthcare than the military in the coming years, because everybody's going to get old, right? And so we have an aging population, just like Japan. If you go to Tokyo now, you can see exactly what we're going to be, which is that a bunch of old people. And that means that there's going to be a huge industry to sell longevity drugs to you. So I just want you to be wary. I don't actually have a longevity drug to sell to you just yet, but I want you to be wary. That's all I'm saying here, okay? So what can we do, right? This goes to building the thing in 10 lines of Python code. What can we do if we don't like have a PhD and we don't work at one of these companies? Well, we could get a job. That's always an option. And there are 20 biotech companies that are hiring right now for people, data scientists, bioinformatics, people in the space. New Limit is one I was talking with one of the people here earlier about, my friend Brian Armstrong, who started Coinbase, and now he's working on that. This is a really cool one. I would highly recommend new limit. Google started Calico 10 years ago. You've got Jeff Bezos spending all his money on, aging. It's a very billionaire thing to do these days, right? So that's one option. But if you're like me and you don't want to necessarily go work for the man and you want to start something of yourself, let's talk about what that looks like. So what I have here is a small little video of what's happening in the cell. And it's actually not going to play because I'm not connected to the Wi-Fi. But I'll explain what's happening.
We'll connect to.
We'll connect, okay.
We can.
This one? Okay. So basically, it's not going to play. So, no, it's not the Wi-Fi. I did something wrong.
Oh,
it's not using.
Describe it.
I'm going to describe it. So if you go to the smallest scale, if you go to YouTube and you type in biological machinery first video, what's happening at the smallest scales when you really zoom in using equipment that was created in the past 30 years? It's a bunch of robots, and they're going on these levers and pulleys and all these complex physical machinery. All of this is happening at this molecular scale. In a picosecond, hundreds of thousands of petabytes of data are being generated, scales that you and I could not even dream of being able to fit into our three-dimensional head. This is multi-dimensional data, hundreds of thousands of dimensions. So all I'm trying to say is that it's super complicated. That being said, as complicated as it is still information. And like all information, it can be organized. And I'm going to try to organize it as best as I've understood it while working on it for you today. One way to think about life at the smallest scales is a stack of layers, just like a software stack in a computer. At the bottom of the stack, you have the genome, the DNA. This is what has the storage, the hard drive. All the information about you is stored in the genome. Now, every disk needs a reader, some way to read the disk. That's what the epigenome is. And that's where you're seeing a lot of the innovation today. Data that's coming from the epigenome tells the body how to turn on and off all of these switches in the genome, all the A's, C's, T's, and G's, when and where to turn them on and off. Now, there's also another layer on top of that. It's called the transcriptome. What the transcriptome does is it translates all of those on-off switches to different sections of the body. And all of those translations need to be turned into applications. There needs to be an app store for the body. And that app store is called the proteome. Proteins are the apps in the body. Proteins, there are thousands of proteins, and each of them does an individual task really well. And one way to monitor all of the actions of these proteins is with a process monitor, just like we would on a computer. What are all the processes happening? And that process monitor is called the metabolome. And that's that metabolic subway map that we saw earlier. That's called the metabolome, which monitors the actions of all these applications, the proteins, that are created through this translation process by the transcriptome that comes from the on-off switches in the epigenome that comes from the hard disk of all biological beings, which is their DNA. Now, that we understand the stack of how biology works, how can we change it? Now, bear with me for a second. So, DNA is a sequence of A's, C's, T's, and G's, right? And in computer science, we learned that computers operate on sequences of ones and zeros. So one thing is certain here, well, two things. Both computers and biology are both computers, right? That's for certain. The other thing is that they both operate on sequences. So there's some similarity there. What if we could convert the sequences on a computer into the sequences of biology? What would that look like? Well, what it would look like is a map of gates, of logic gates, of conditionals. If-then statements. If this happens, do this, and then do this, and then go back and do this. How do we do that? Well, we're going to design a logic gate, and the way that we're going to design this is a library called Cello. It's called Cello on GitHub, and I'll talk about it more in a second. But Cello allows for genetic circuit design in Python. And once we design this, and there's a couple examples, this is one of them. Once we design this gate, and we're going to basically design it to create a new type of small molecule drug that is going to pull from a database of what are called ligands. And ligands are sequences of molecules. They come from different drug databases. And we're going to tell it what machine to run on as a parameter, the CPU number. We're going to run it on our CPU, because it only needs a CPU, really, to generate all these different variations of the base ligand. We're going to set these 4 different parameters: a charge, multiplicity, solvent, and then where to save it. Now, these 3 parameters can be changed. We can play around with them, and we can use an optimization strategy, namely, drumroll please, AI. We can use AI. That's where AI comes in. Remember, this data is way too complex for you and I to understand, but that's where AI comes in. And so AI is going to help us understand which parameters to use to generate the optimal molecule, given this ligand database. And this is all going to construct a molecule using a series of logic gates that are written in this cello programming language. Now, this should be confusing, which is why I have one more example from a different angle to give you a different view of it and a more holistic view of what we're trying to do here. Now, I said that cello was one library, but there's 2 other libraries that I want you to know about. The first one is called Coral, and that's even more recent. The second one is called BioPython. Actually, great name, by the way. They should have, that's a great name. So what we can do with these libraries, I talked about cello. Another one is Coral, is once we import that library, we can create what's called a primer. And the first thing that people do in a molecular biology lab is create what's called a primer. And what a primer can do is it can, inhibit or, say, amplify a certain gene. And in this case, it's going to amplify this gene. Basically, it's going to make it so that it's going to be activated in whatever organism that we're going to insert this into. And it's going to read DNA from a file that was sequenced from some organism, maybe a human, and then it's going to show you the sequence of A's, C's, T's, and G's, the code of biology, right? And so once we have that, we can run a design process using Coral on that data to create a bunch of variations. And we can just test them out. And right now, the process of testing it out takes a decade, right? The drug design process. We have clinical trials, phase 1, 2, 3. And it's a huge, huge choker on the drug discovery process. It slows it down way more than it needs to, which is why quantum computing is very exciting. And Richard Feynman said in 1965 that the way we're going to simulate quantum mechanics is through quantum computing. And imagine if we could simulate this on a biological organism that lived on a digital machine, essentially simulating a drug trial on a computer in silico, avoiding the need for the entire clinical trial process and being able to print a drug from idea to patient in, N of 1 a day. That's the future that I foresee with this technology. But we need a lot more attention on it for us to get there. Now, let's say we have this algorithm. You and I, we've done this. We've got this 10-line of Python code. Great. Where are we going to deploy this thing? Well, it turns out Amazon, of all people, they have a health omics platform. When did this happen? It happened 2 years ago, under everybody's nose. It's called AWS Health Omics. Transform genomic, transcriptomic, and other omics data into insights. Very cool stuff. I really already want to get started with this, don't you? Very cool stuff. So that's one way that we could deploy this. Amazon should pay me, right? That's one way that we could deploy this. But there's other ways as well. We could deploy it to our own server. We could create a paid subscription service for omics data because of how valuable it is, which brings me to me selling my genome as an NFT, which is kind of the gimmick for this talk, right? This is actually a really important moment in the history of Web3 because, well, first of all, somebody bought my genome for 1 ETH, and they resold it twice. And they flipped my genome for 3 ETH. So somebody made money, and then made money, and then made money again. Now, if I would have uploaded my genome to any normal website, forget blockchain. Blockchain's a scam, I'm not going to do that. Someone would have resold it, and I wouldn't have made a dime. But because it was encoded on this public transnational ledger, Ethereum's blockchain, that nobody controls, every time it was resold, guess who made a percentage cut of that pretty penny? Me. And guess whose data it was in the first place? Mine. So don't you think I deserve to get paid for that data? And I think you do too. The way to make that happen without one entity controlling it is through some tool that nobody controls. 23andMe is the biggest American company, that's where I sequence my genome, that has the biggest database, and it's already being sold to the highest bidder internationally, right? So we have a huge problem with health data in this country, and centralized solutions are not cutting it, right? They're just not cutting it. And so we need a way for people to, first of all, make money. And one of the things that you and I, all of us have, that no robot or machine has, is our biological data. In fact, it is our most valuable asset. And we should make money from that. We should not just give that away for free. And the way to have a type of digital ownership where we don't have to become a serf to some server owner is to take that data and create a patent that lives across countries. That patent is called an NFT. So while everybody else was losing money with NFTs, biodata was making money. So I think that this is really the black swan for where blockchain meets AI meets science. We're going to see this convergence of 3 different technologies. We're going to see genomics data. We're going to see the blockchain to monetize this data for ambitious researchers who live outside the bounds of academia, who want to push forward the space, who are willing to say no to the existing health status quo and want to create new innovations. Those are the people who are going to make money from this. And I look forward to helping them on that journey. And I also want to say that gene therapies, that is designing information for living organisms with computers, is not limited to just ending disease. We can create new types of abundant food. We can create crops that have 100x yield. Most of the fruits and vegetables that we eat today are already genetically modified, right? They didn't exist even 100 years ago. Human enhancement, right? And this is what we were talking about the enhanced Olympics. So that's already a topic. Clean energy, biofuel, things like this. And I also want to just end the talk on a very kind of out there idea to end it with. And that is, plants are computers. And if you've ever had a plant, that might not have been apparent to you, but I grew tomatoes for a little bit, and they were my little computers, and I really loved them because they were able to take sunlight and convert that into energy. And I did a little bit of research onto how they were able to do that, and it turns out that they use a form of quantum superposition to perform photosynthesis. And that means that that's a form of computing, in a sense. Biology, like we talked about, is computing. And what if we could repurpose that computing for not just individual actions for regulation and dysregulation inside of an organism, but perhaps for everything else that we want to do with computers? Why do we have to have data centers that look like the way they do? What if data centers look like greenhouses? That would be much more aligned with how the Earth is. And in that way, we're using AI not just for the longevity of humanity, but for Earth itself. So I think that synthetic biology is probably the most exciting topic for the next decade. It is where AI is headed. And if you are interested, it's time to build. I'm a part of, yes, I'm a YouTuber, and I'm CEO of Wager GPT, and we're building sports betting AI. But this is really my long-term goal. And I'm a part of a venture capital firm as well, Modi VC. I don't talk about it too much, but I'm not very active. But any excellent founders in this space, I just hand over to them, and we're going to get you funded if you've got a great idea. And I'm happy to do that. And yeah, thank you, everybody. Any questions?
How much did you win yesterday?
My members won a lot. I actually didn't bet, but they were happy with the Chiefs one. Yeah. Great.
So I guess what's interesting is the pitch on gene therapy. The analogy you make for the computer is like, I guess you don't go right into the hardware, to update or find out optimizations in the iPhone. So when you go back to the omics tab, like, why focus on the gene versus stuff that's more of the software higher level component? If that question.
This one?
Yeah. Like, why focus on genomics versus the ones at the top?
No, this is so much of the opportunity lies right here. I think genomics was really just the first data set that we got, right? Watson found DNA, but these are new fields. Proteomics is like one of the hottest topics right now in molecular biology, just from like a sideline, looking in the subreddit and stuff like that.
You mentioned a couple of solutions or like studies or whatever where like, oh, this drug was meant to do this, but it turns out it also made people live longer. And you also in an earlier slide were like, here, look at all these arrows, all of these things are caused by aging. So do you think that some of those drugs were capturing like a small part of the solution to aging, and then like we're kind of like we're only seeing like a part
mean, I have no idea, but if I were to make a guess, like based on what I've seen in AI, so much of the progress that's happened has been from taking away ideas rather than adding them. For example, attention is all you need was a seminal paper, 2017, right? All these other recurrent networks, all these different schemes to get data to reverse back into itself, and none of it mattered. You just remove all of that, and attention, which was just a mechanism of deciding what parts of the input were most relevant, that's all you needed. So I suspect something similar is in biology, where it's just something, of course it would have been, right? That's how turtles do it, or this animal does it. But I don't know.
When you sold your NFT, did you sell how did that work? Did you put did you put your genome on chain, or did you get the rights to it? Like, how did it work logistic-wise?
That's a great question. So like I said, it's a .txt file with ACs, Ts, and Gs. What I did was I uploaded it to what's called the Interplanetary File System, IPFS. And what IPFS is it's a database layer for blockchains. And so you can basically think of IPFS as a blockchain, but for storage. When you upload something to IPFS, it's not stored in one computer, it's stored in a network of computers. So your data is split into many parts, and it's stored in bits and pieces across the world in what are called miners for IPFS. And they're paid in what's called Filecoin. But you don't have to interface with that. It's kind of complicated, but it's actually not. Step 1, upload data to IPFS. Step 2, imprint NFT on that data with Ethereum. And that process is just a transaction with the ERC 721 signature, as all NFTs have.
So, but then anyone who went to OpenSea could see that IPFS upload to your genomic data.
No, so you had to unlock that NFT by purchasing it, and then you got to see the link.
Oh, okay, got it. And do you worry about previous owners when you sell it? You could copy it, but is that not a worry?
You would think it's a worry, but they would first have to buy it, and if they decided to buy it, and then they could technically find a way around it, but there's schemes of basically blotting the data unless it has like an encoding, like a key signature, basically a hashing algorithm. I didn't get that far.
Or you can watermark it.
Yeah, basically, that's a better word for it. Yeah.
Got it. So, okay, and it's not as big of a deal if only like 10 people have it. They're probably not going to... Or, I mean, maybe just owning that genomic, like, many people have owned the Mona Lisa, but still owning the one that, like, is like, the actual one is the most important thing versus, like, making photocopies of it.
Yeah, and then most importantly, getting independent scientists to make money. Yeah. I think that's the primary motivation for me in this space. That's what matters the most, because it's really hard, to get funding for your crazy scientific ideas. And the more comfortable we make scientists with basically blockchains, the easier it's going to be for them to get funding. Because there's a lot of money to be made here still, especially for science. It just makes a lot of sense to just get on board with this. And if your project works, your token's going to accrue in value as well. Actually, there's a great example of this, like a legit example, out of Coinbase. It's called Sci-Hub. It's essentially like Brian Armstrong's science token project. It's been going on for 4 years.
Research Hub. Sci-Hub is where all the papers are uploaded.
Right,
they were a speaker here once.
Yeah. Research Hub is a great example. Yeah. They do it well.
How did you need a prescription or some sort of thing to get your blood drawn, or how did that work logistically?
Yeah, so I just went to InsideTracker.com and then I bought their premium plan. It's way too expensive, literally, like, and it's like $400. And then I went to Quest, not nutrition, but I think it's Quest Nutrition. Diagnostics. And then, I scheduled a blood test like a Walgreens. And it sucked. Like, I don't like taking blood tests, right? It's like a lot of blood, and the lady didn't care. That's even worse. And it's a lot too, because it's 42 biomarkers. Because they got their premiums, it's like 4 vials about that big. It's uncomfortable, but it's also really exciting. Like, nothing happened, and then I got my data in 5 days after I got the blood test, and it was just basically a chart of all these things. And honestly, Insight Tracker's like whole AI thing is a joke. I don't even use that. It's totally wrong. I just took the data, downloaded it, and then with Python just visualized it different ways, and it's just really interesting for me.
You can also probably just upload it to ChatGPT, and it would probably tell you exactly what a doctor would say.
That's exactly right. I think you can do it for like $100, just without the Insight Tracker part. Just go to Quest, get a blood test, and you can do it with ChatGPT. Exactly. That's the most cost-effective way. Like, how do I reverse my age given this?
Yeah.
Give me like the foods and diet, exercise. Oh, well, what do? Here. Okay, here's what you do.
Yeah, exactly.
Yes.
Can you explain for the Insight Tracker the algorithm that was used to calculate the amount of years, plus or minus, that was generated? And I think you mentioned that it was based on a standard of 18 years old. How was that calculated? Was that an aggregation of data, or what is that based on?
Yeah, so what they use, they have a proprietary algorithm for computing that, and it's, they call it the inner age score, and they recently updated it to 2.0, and they updated that score between me taking this test and this test. So it's actually unclear if it was even me or their score changed, right? But I don't like to say that too often. I guess I'm saying that now, right? So, but my point in saying that is that is important, is that it's proprietary. So we can't actually know how accurate this is, which goes back to show how important it is to have an open source algorithm that if you're going to take a blood test, you have to know how it works. And the most popular open source clock, and there's several what are called aging clocks that you can use to compute your age given your biological data. The most popular one is called the Horvath clock by Steve Horvath out of UCLA. And yeah, the Horvath clock is the main one, and that's the one I would probably use. Yeah.
When you do the computing with the data, and because you are using generative AI, CPU is not enough. You need to now use GPU. And for these special platforms, where you can move the data from silos to pipelines, you can visualize, you can store it, and they have the very powerful platform with a big GPU on it. I'm using it actually with that.
Well, it's also, no, I mean, GPUs are the way. This was just like a smaller example. But anything serious needs to be done on a GPU, for sure. Or one of Sam Altman's, proprietary $7 trillion processors that he's about to build. Yes?
What are some DeSci applications you want to see built? And then, do you think there is some product or like potential solution to acquire user need or to get users' data or like patients' data more open source and then upload it and then give each patient ownership of the data? Like what kind or how should that application look like? Or are there any other angles besides this one?
Yeah, so I think first things first is we all need to be students of history, right? So I was working at Polygon in 2021, which is top 5 cryptocurrency at the time, before FTX happened, right? So I was working there as head of developer education, and it was a lot of fun. I got a lot of insight. But what we saw was a complete failure in what was called the health to earn space. You saw these startups that were saying like, yeah, you're going to earn this coin if you get healthy, if you play this health game. And the biggest example was Sweatcoin. It was called Sweatcoin. And Sweatcoin was around for about 3 years, and it is exactly how it sounds in that you earn Sweatcoin if you run. And then you could exchange Sweatcoin for different gifts in a gift shop. And actually, Sweatcoin did accrue in value, and people were making some money. But then, for whatever reason, the tokenomics, it wasn't sustainable, which is almost always the case. The tokenomics is not sustainable in the long run. And I'm just waiting for the crew that's able to actually make it work. Now, who did make it work? OpenSea kind of made it work. OpenSea is kind of this hybrid, centralized, decentralized model where there is a dedicated team, and they are, in one office, but they don't own the art, the NFTs that are posted on their platform. They don't own my genome, but they helped me list it, and they were a discovery service for other decentralized buyers. So I think that is something that is a signal, a marketplace for health data. It's something that I've actually thought about for years, but
I've seen 8 projects like within this space.
You've seen 8?
Yeah, like 8, or maybe 5. I forgot. I lost count. But then none of them are super successful.
Yeah, I think it's really hard to make the tokenomics work for this.
No, they don't have tokenomics. They're just like trying to give the I really have seen 5 projects maybe in this, trying to solve this problem, but none of them are super big and complete so far. And then there's maybe 3 projects trying to build decentralized clinical trial products, and then none of them are successful yet.
Well, they say that, seasoned biotech investors, unlike me, they say that it takes 20 years for a biotech startup to be successful. That's a centralized biotech. It is. I agree. It should be faster. I totally agree with that. There's always one golden goose in a pile of crap, right? Like Coinbase is our shining American example of a crypto exchange that is reputable, that you can trust with your money. It's not going to collapse like FTX did. And they're fully regulated, right? So, and everybody else was a scammer at the end of the day, right? FTX, everybody else, Gemini, everybody else fell. So I think that's what's going to be the case with biohealth data. There's going to be one company that does it well. If I knew the answer, I'd be working on it. I don't know the answer yet.
I think there should be a big major bio player who actually I don't know, or just a big crypto player who can actually acquire the bio part, or the bio company that can work with the rest of the people.
Yeah. No, I mean, Bitcoin is going up right now, so people are coming back to crypto as it always does. The 10th cycle. Here we go. Party's on.
Any other questions? Well, thank you. That was an amazing talk. Fantastic. And yeah, just enjoy. Thank you all for coming out. We have the other one on February 22nd with the Enhanced Games president of the Enhanced Games, Aaron. Also, fun fact about Aaron. I don't know if any of you guys followed the Hulk Hogan Gawker situation with Peter Thiel, but he was actually the one that came up with that idea to shut Gawker out of business. He was the guy that actually pitched it to Peter Thiel to fund it, to find Hulk Hogan, to sue Gawker to get back at Gawker for outing Peter Thiel's gay. So very roundabout, but he came up with that idea, so he's a very interesting person. And you guys are all welcome to come. So thank you guys for showing me up.
Wow, shameless yoga movement here.
Cool.
Thank you for having me. Yeah, thanks, Michael. Really appreciate it.
Nice. Would you like?
Nice stuff.
Pop up, right?
Yeah. I can walk out and I have my 2 sentences for the DeSci people. Every day. You only give so much. You have to start slowly. Give so much. I gotta start with a statement. What? I started so. Yeah, we should go to the next one. I heard about
I might consider it.
It was fun, right? Pablo's DJing the opening party. Pablo's DJing Best Longevity Plus.
This is more based on
How much such? It's really